Welcome to ASAP Science, Tracy Edition. Today's topic is DNA replication. The three stages of DNA replication are initiation, elongation, and edits. To understand the basics of DNA replication, we need to look at the structure of deoxyribose. Deoxyribose has 5 carbons. So as it's going up, we go from 3 carbon to 5 carbon, and as it's going down, we go to 5 carbon to 3 carbon. So during DNA replication, remember this phrase: read 35 pages and write 53 pages.
This is an image of DNA before it splits, and the reason it splits is due to a protein called helicase. Okay, so first we're going to do initiation, in which the first part of initiation is forming this replication fork, and replication fork refers to this little like tumor looking thing, and that's when the DNA separates. So our good friend helicase, labeled by that red triangle, is the one who actually separates this. As you can see, it's moving towards the 3' area or the 5' area, depending on which strand you're looking at. And they have the help from topoisomerase gyrase, which is the TG, the blue circles, and those relieve the stress from when it separates, because actually unwinding and separating it creates a lot of stress on the actual DNA. And you may be wondering, how do these strands stay apart during the actual replication? Well, that's when our friends, the single-stranded binding proteins, these little black dots on the outside of the DNA strands, or SSBP for short, and they keep the two DNA strands apart in order for replication to actually occur. So now that we have the replication fork form, we actually need to start getting the other strand of DNA. And this starts off first with a protein called primase, which drops off RNA that makes a landing zone for the future DNA polymerase 3, which is represented by the blue. Again, the blue is DNA polymerase 3, in which it creates complementary bases depending on the DNA it's reading. So if the original DNA says A, then the polymerase puts T. And an important thing to remember is that it reads from the 3' to the 5', and it writes from the 5' to the 3'. And primase, its job is to drop off the RNA primers, emphasis on RNA, to make a landing zone for actual DNA polymerase 3. Whereas DNA polymerase 3 starts at the landing zone, which is indicated by the pink, and again adds complementary bases. This is the start of the actual DNA process, and this image is actually an example of the leading strand, which means that DNA polymerase is moving in the same direction as the helicase, meaning it's moving faster and only has to put 1 primer. This is different from the lagging strand, which will be discussed later in the video. For those who don't know, a primer is a chunk of 3 RNA bases complementary of the DNA. Complementary meaning if the DNA is A, then its complement would be T, because adenine and thymine are complements of each other, and guanine and cytosine are also complements of each other. After elongation becomes the step of edits, and this prevents any mistakes from being made within the replication process. So first, DNA polymerase 1 comes and swaps the RNA primer for DNA complementary pairs. Now this is important because last time I checked, our DNA doesn't have any RNA in it, and so the DNA polymerase 1, not to be confused with polymerase 3, takes out the previous RNA primer and swaps it for DNA. That's only one of its functions. Its second function acts as a proofreader to prevent any mistakes from occurring within the process of replication. For example, if 2 A's were together, then it would fix it to be an AT or a TA. Think of it as spell check on Microsoft Word, but for your DNA. And they do this by breaking the backbone of the RNA primers to repair it or to swap it. And then we get our good friend ligase. He's the doctor of the group. And what he does is, if you notice, there is a gap between the newly placed DNA from DNA polymerase 1 and the DNA placed by DNA polymerase 3. And so what ligase does is that it closes that gap between them. Or in other words, if you want to be scientific, it joins the Okazaki fragments, and it's named Okazaki based off the scientist, because scientists love to name stuff after themselves. And as you can see, I'm doing an example in the corner in which it shows what ligase does as it's connecting two fragments. This entire video so far has been focusing on only one strand of DNA replication, in which it is called the leading strand. The reason it is called the leading strand is because it's following the same direction as helicase is moving, from the 3' to the 5' side. So it's following the rule of read 35 pages and write 53 pages. So now we will be focusing on the lagging strand, which is opposite of the leading strand. So like always, primase will drop off an RNA primer, and DNA polymerase will begin to follow the rule of read from 3' to 5' and write from 5' to 3'. However, it is because of this rule that the lagging strand moves slower, as it is not moving in the same direction as the helicase. This means that more RNA primers would need to be dropped off in order to keep up with the leading strand. So, as helicase opens up more DNA, every 100 bases or so, a new RNA primer would be dropped off, and new DNA polymerase 3 would make new DNA going from 3, or following the rule of reading 3' to 5' and writing from 5' to 3'. So this is important because the lagging strand has more RNA primers, which means that there's more breaks. So when DNA polymerase 1 comes and replaces those primers with actual DNA, there's going to be more fragments, more Okazaki fragments. And that's where ligase is super important, as it'll repair those by binding those two Okazaki fragments together in order to make it seem seamless. And this is the reason why the lagging strand is so much slower, as it needs more time for the Okazaki fragments to form and actually place all the pieces necessary to make it. Okay, so if you're having some trouble memorizing all of these proteins, here's some pictures and descriptions to help you. So first, let's go with the first one, the knife, aka helicase. So we name it the knife because it's the one that's cutting open and splitting apart the DNA to make the replication form, such as a knife cuts through any object basically that's squishy. So next we have the divorcer. So this one's funny because it's called the single-stranded binding proteins, and if you remember, those are the ones that basically keep the two strands apart. So the reason why it's called divorcer is because these single proteins have been through a divorce, and they're so desperate that they latch onto the single strand of DNA and will never let it go because it's so lonely and it wants someone in their lives. So next we have the stress relievers, aka the topoisomerase gyrase. And the reason why they're called that is because, if you recall, they alleviate any stress caused by helicase when it's separating and unwinding the DNA. So those are for the initial, the three buddies. So next, we have the landing zone expert, aka primase. And the reason why it's boxes and crates is because it does provide the landing zone, aka the origin point for where the DNA replication will actually occur, in the form of RNA primers. Its partner in crime is our DNA builder, DNA polymerase 3. And the reason why it's a jackhammer with what looks like railroad tracks, but it's actually supposed to be DNA. But what it does, it acts like a jackhammer, and it basically hammers down, bam, 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 bases, complementary bases, to be more specific. And then we have its wannabe, the swapper, aka the DNA polymerase. So the reason why it's called the swapper is because it swaps the RNA primers from the primase for actual DNA so that it would match and there would be no RNA within our DNA. And then last but not least, and the most important actually, is our Dr. Ligase. What Dr. Ligase does is he heals and makes sure there's no mistakes within our newly replicated DNA. And he patches up the holes between the Okazaki fragments that were caused by DNA polymerase when it was switching out the primers from primase. So Dr. Ligase is actually pretty important, as he acts like the Microsoft Word for DNA, as he prevents any mistakes